Hi everyone. So one of the things that you've all asked me to work on with you all is how to improve your answers for the required practical questions that come up in your exam papers. A few things you need to remember before we begin. Firstly, when you're talking about mass or volume, we need to refer to those terms as opposed to using amount. Secondly, we need to remember the definition of two keywords that are often forgotten, independent and dependent variable. So independent is what you're changing and the dependent variable is what you measure. So try not to mix those two things up. Now, when you're suggesting improvements, try to think about improving the method rather than the equipment initially. Now, we're going to talk about when you talk about equipment a bit later. Validity means, does the experiment actually test what it sets out to test? And reliable is now an outdated term. Your exam papers are going to be using terms of repeatability or reproducibility. Again, we're going to go through the definition of these terms and how we use them a bit later. So we're going to begin by going through some key terms. The first one is accuracy. Now, when an exam question talks about accuracy, it's talking about how close your values are to the true value. Second one, or the second two that we're going to go into, are linked to errors. So you've got two types of errors. You've got random error and systematic error. Now, a random error is when your data is spread about the true value due to the results varying in an unpredictable way from one measurement to the next. Now, random errors are present when any measurement is made and cannot be corrected. The effect of random errors can be reduced by making more measurements and calculating a new mean. The second type of error is a systematic error. Now, these cause readings to differ from the true value by a consistent amount each time a measurement is made. Sources of systematic error can include the environment, methods of observation, or instruments used. Now, systematic errors cannot be dealt with by simple repeats. If a systematic error is suspected, the data collection should be repeated using a different technique or different set of equipment. Precision. Now, the precision is different to the accuracy. Precise measurements are one in which there's very little spread around the mean. So that's the crucial point here. Now, precision depends only on the extent of random errors. It does not link to the true value. So that's where accuracy and precision vary. Accuracy is linked to, does it actually meet your true value? And precision is based around the mean, which might not be anywhere near your true value. Repeatable. A measurement is repeatable if the original experimenter repeats the investigation using the same method and equipment and still obtains the same results. Whereas reproducible is if the investigation is repeated by another person. So here's the key difference between repeatable and reproducible. So a different person must be doing the investigation by using different equipment or techniques and still obtains the same results. Okay, so that's repeatable and reproducible. Last two are valid. Okay, so validity. Now, this is when the procedure is suitable to the actual investigation and the questions being asked. So, does your method actually link to your hypothesis? That's what you need to look at when you're asked about validity. And finally, a valid conclusion. Now, a valid conclusion needs to be supported by valid data. Now, this is obtained from an appropriate experimental design and based on sound reasoning, again, linking right back to your hypothesis. Now, the first type of exam question we're going to look at is when they are actually asking you to compile a method linked to a required practical. So I'm going to go through with you a few things that you need to include depending on what's appropriate and also the number of marks that they're asking for. So sometimes these come up as 6-mark questions, sometimes they come up as 3 or 4-mark questions. So obviously based on the number of marks, you need to hit the certain number of marking points. Now, a few things you need to include are a range of values that you're actually going to be testing with the correct units. Of course, you need to go back to the independent and dependent variables. Again, you don't need to mention them by name, but you need to include what they are. The control variables, okay, so that will link directly to you having actual valid results. A risk assessment, again, it doesn't need to be like a formal table, just a few things that could be hazardous in your experiment that you might need to control. Repeats, okay, again, you need repeats to identify any anomalous results and then to calculate your mean and standard deviation, which links directly to you using statistical tests. So including a statistical test, especially in 6-mark questions, would bump your marks up. And lastly, an expectation of correct results should be included at the end if they ask. Now, longer answer questions in your exam papers will ask you to evaluate a method or a statement. So they'll give you an experiment and ask you to evaluate that method. And in that answer, you must include both for and against arguments. That then must link to some reasons and data, okay, to support your answer. And of course, citing the correct units where appropriate. The first thing you want to do is actually look at the data you've been given for that experiment that was conducted, okay? And look at the data set, whether that's in a table or in a graph, okay? Now, are there any labels missing? Are there any units missing? And that's the first kind of things you need to include in your answer. Moving on, you need to then have a look at the text part of your answer. So analysing the text part would mean that you need to highlight the key terms in that answer, okay? So you want to go and identify the control variables, independent variables, etc. Now, of course, if there's any control variables missing, those are things that would then form part of your against argument, okay? You want to look at repeatability, okay? So are there repeats in the experiment? Has there been any calculations conducted? Can you then calculate the mean if there are no repeats, okay? And that would then link directly to your accuracy, okay? So you want to look at your specific volumes, your mass, the concentrations, okay? If it's an answer based on drawings, okay, then you need to look at whether those drawings are done incorrectly or not. Sometimes the calculations they've done are wrong, okay? So you need to recalculate them to see whether then that would form part of your argument, okay? Lastly, correlation does not imply causation. So that again is crucial for you to look at the control variables. Are there any other variables that may actually be causing those results? Okay, and that again could be that the method's been done incorrectly, wrong. You know, for example, the wrong color filter on a colorimeter. Okay. Now, when you're looking at for arguments, okay, you can of course use anything opposing any of these. Okay, and ultimately you want to look at, does it follow the correct trend? Okay, and if it does, that's what you want to include in your answer. Okay, if some parts of the method are correct, that forms part of your answer. Okay, and like I said, you want to finish off by concluding your findings. Okay, and that's how you can get your top marks for an evaluating a method question. So far, we've looked at longer answer questions. We're going to focus a little bit on shorter answer questions on required practicals, and they tend to link to how you can possibly improve the validity or the reproducibility of the experiment that they've spoken about. Now, a few things you can do are listed below. Okay. Now, when it comes to any of these statements, you want to talk about increasing range of values tested. So have a look at the data that they've tested and can you increase that range? Check the variables. Can you control more variables? Okay. How many repetitions are there? Try and include more repetition of the data set. And have control experiments. Okay, there are a few more listed here that you can read through. We're now going to go through a few practice questions, and I'm going to talk you through how you can answer them. So the first question links to a procedure that you would have done in lesson. Okay, so biological processes can be investigated using models. Okay, the effect of cell size and diffusion can be investigated using cubes of agar jelly to represent cells of different sizes. Now, the procedure is written there. Okay, what you need to do is suggest how the original procedure could be modified in order to improve the accuracy. Now, this is a 1-mark question. The first thing you want to do is obviously recap the definition of accuracy. Okay, and that is a measurement result is considered accurate if it is judged to be close to the true value. So how are you going to improve how close your data is to the true value? Okay, so if we have a look at the steps that were taken, okay, you've got 5 different sizes of cubes are taken. These were placed in a beaker containing hydrochloric acid. After 2 minutes, the cubes are removed, rinsed with distilled water and blotted dry. Acid absorbed at the outside continued diffusing towards the center of the blocks, and the time taken was recorded. Okay, so what part of this question talks about accuracy and your true value? Okay, once you've got your data set, what could you do to then improve how close your answer is to the true value. Okay, so right, so looking at the procedure, we can see the one thing that wasn't controlled was the different sizes of cubes. Okay, so in order to improve the accuracy and make sure that the data set is linked as close as possible to your true value, you would need to control the sizes of those cubes. Okay, so to get that 1 mark, all you need to do here is test cubes of known length between 10 and 20 millimeters. So that was a simple 1 mark question linked to accuracy. A longer question, this time linked to validity. Okay, here you've got an experimental procedure written up, and the question is, suggest how the procedure could be improved so that a valid comparison could be made. Okay, so the key term here is valid. I'm going to recap the definition of valid. Okay, so suitability of the investigative procedure to answer the question being asked. Okay, so you want to, does the actual procedure link to your hypothesis? Okay, so the question is, on a biology field trip, some students carried out a survey of butterfly species in 2 areas of heathland. One part of the heathland was used regularly by walkers, while the other had been deliberately fenced off by the National Park Authority to promote biodiversity. So area 1 was accessible by walkers and area 2 was fenced off. On 2 different mornings in June, the students walked along a transect in each area 4 times at 30-minute intervals and recorded every butterfly sighting. The aim of the survey was to compare the biodiversity of butterfly species in the 2 areas. Okay, so in order to improve the validity, we need to make sure that all the variables are controlled except one, which is of course your independent variable. So we're going to look through the actual method and make sure that there's only one variable. Okay. We also need to mention the control and independent variables in our answer. Okay. So for this, we can see that there are some of the variables that haven't been considered. Okay. For example, weather conditions. Okay, that's not listed in your answer. Neither is the fact that you might be examining the butterfly species different times of the year or different times of the day, and also different parts of the actual area. Okay, so in order to improve the validity, we want more transects in different parts of area 1 and 2. Okay, you want to collect the actual data at different times of the year or day or throughout different weather conditions. You also need a method to ensure that individuals aren't counted again. Okay, and so therefore you need to employ a method of capturing those individuals. And finally, you need to make sure that you're actually identifying those species correctly. Okay, so that's it for the video. I hope you found it useful and found some techniques to help improve your answers to required practical questions. If you have a go at some questions now and give me some feedback, and I can help you with any further questions that you may have.